Hello, everyone. Welcome to today's live broadcast, Manipulating the Microbiome to Prevent and Treat Infection. I am Robert Castellanos of LabRoots, and I'll be your moderator for today's event. LabRoots is the leading scientific social networking site, and we are proud to bring you this interactive web seminar. For more information, please visit labroots.com. This is going to be an interactive presentation. We want to hear from you. And this means you can ask questions throughout the presentation by selecting the green Q&A button at the lower left-hand corner of the screen. And we'll make sure to answer as many of those questions as possible. You can enlarge the slide window by clicking on the screen icon at the lower right-hand corner of the slide window. If you can't hear or this presentation seems not to be functioning properly, please let us know by clicking on the support button at the top right or the Q&A button in the lower left. This is an educational webinar and thus offers free continuing educational credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would like to now introduce today's speaker, Dr. Dan Diekem. Dr. Diekem is the Director of the Division of Infectious Diseases at the University of Iowa Carver College of Medicine, Associate Director of the University of Iowa Clinical Microbiology Laboratory, the Associate Hospital Epidemiologist at the University of Iowa Healthcare. He has served on national committees that have established standards in clinical microbiology and infection prevention, including the CDC's Healthcare Infection Control Practices Advisory Committee, of which he is currently the co-chair. And without further ado, I would like to now introduce the doctor.
All right, thank you all for tuning in for this webinar. It's my pleasure to be able to chat with you a bit about how we are currently manipulating the human microbiome in efforts to both prevent and treat infection. So I'm just going to launch right in here. As most of you already know, humans are basically walking ecosystems of microbial life. If you count up all the bacterial, fungal, and viral cells that live on or in human beings, they outnumber human cells tenfold. And the genetic material as well, much more abundant microbial genetic material living in and on us than human genetic material. Most of these organisms are bacterial, and those are the ones that I'll be talking about most today in terms of the ways that we approach microbiome manipulation to prevent infection. So one of the most important concepts of the human microbiota is that it varies quite dramatically from one body site to another. If you look at this slide, for example, for the gastrointestinal tract, you see it's really overwhelmingly consists of firmicutes and bacteroidetes, whereas for skin microbiota, you see a lot more actinobacteria, and as you go to the different parts of the body, you see different phyla that are predominant. There are other factors associated with variation of microbiota from person to person, a lot of host factors, environmental factors, including diet, as well as treatment factors like exposure to antimicrobial agents, which obviously alters the microbiota significantly. Now, as was alluded to on the last slide, when we talk about bacterial organisms in the microbiome and microbiota, 99% or more of these are beneficial. If they weren't, very few of us would still be alive. It's only rarely that one of these organisms acts as a pathogen. So the distinction between a commensal or a normal part of our microbiota and a pathogen often just depends on the situation, and an organism that may normally be a commensal can become pathogenic when certain things occur, such as suppression of the immune system or if a barrier, skin barrier, mucous membrane barrier is impaired with things like a central venous catheter, chemotherapy causing mucositis, etc. The other way that a commensal can become a pathogen, and we'll discuss this in the context of Clostridium difficile, is if a normal healthy microbiota is disrupted significantly, that can create an opportunity for part of what is usually commensal flora to become pathogenic. The sites that I'm going to focus on in this talk are the areas where we're currently using manipulation of microbiota to prevent or treat infection, and those are circled on this slide: the nares or nostril, skin, and the gastrointestinal tract. So let's start with the nose. So this graph is from a paper by Bassis and colleagues in Microbiome from a couple of years ago, and it's a metagenomic study of the healthy adult human nasal cavity. They took 10 adults and they looked at the organisms that were present in the nasal cavity compared with the buccal mucosa and the tongue. And really the only point I want to make from these data is that if you look at the aqua color, which represents staphylococci, you see that that really is only present in the nasal cavity. You see very little or none of it in the buccal mucosa or the tongue, because it appears that, and we've learned that, the nares of humans is a niche for staphylococcal organisms, particularly Staph aureus. Anywhere from 20 to 30% of humans will carry Staph aureus in the nose at any one time. So why is this important? It's important because Staph aureus, aside from being a commensal living in the noses of 20 to 30% of humans, is also a very virulent human pathogen and a significant contributor to human disease. Staph aureus causes up to 1/4 of all bloodstream infections, 30% of all cases of endocarditis, 1/5 of all surgical site infections, and over 1/2 of all bone and joint infections are due to Staph aureus. It's also important to recognize nasal carriage of staph because it appears that those of us who carry Staph aureus in our noses do have a greater risk of infection under certain circumstances. For surgical site infections, this was first reported in the New England Journal of Medicine in 1959 by Weinstein, showing that those who had Staph aureus in their noses were more likely to get Staph aureus surgical site infections. Other populations are also at increased risk listed here: dialysis, HIV population, IV drug users, ICU patients, and those who are recently colonized with Staph aureus in various studies have all been identified as being at higher risk for Staph aureus infection. Furthermore, when these organisms are subjected to molecular typing, study after study has shown that the infecting isolate, the one that ends up being found in the surgical wound or in the bloodstream, is identical to the isolate that was found colonizing the nose in advance of infection. So what is the increase in risk associated with Staph aureus nasal carriage? Well, this is just 3 of the studies that have looked at this, and you can see that in continuous ambulatory peritoneal dialysis patients, cardiac surgery patients, and hemodialysis patients, anywhere from a 3-fold to an almost 10-fold increased risk for Staph aureus infection can be found among those who are colonized with Staph aureus, mostly in the nose. I mean, I should note that staph can also colonize the throat and the GI tract, but most of the published data looks at nasal carriage, which is the site where it's found most frequently. So this leads to an obvious intervention, which is, okay, if nasal carriers are at increased risk of infection, what if we try to eradicate nasal carriage or suppress nasal carriage by looking or seeking out those individuals who have carriage and then trying to decolonize them, using usually bupirosin in combination with chlorhexidine body washes to prevent or eradicate organisms that transiently colonize the skin, often from the nose. So there are a lot of studies that have looked at whether screening and decolonization of Staph aureus reduces infection rates. Many of these are observational before-after studies. I'm showing you this study because it's one of the few multi-center randomized controlled trials by Bodie et al., published in the New England Journal of Medicine now almost 6 years ago. What these authors or investigators did is that they screened hospital admissions for Staph aureus using PCR to look for nasal carriage, and then those who were carrying the organism were given chlorhexidine with mupirocin in the nose twice a day for 5 days versus normal management. The primary outcome in this study was hospital onset Staph aureus infections, and since most of the patients in this study were surgical patients, most of the infections were surgical site infections. So here are the results of this study. This group of investigators was able to demonstrate an almost 60% reduction in hospital onset Staph aureus infections through the use of mupirocin and chlorhexidine among Staph aureus carriers. And if you look just at some of the most serious of Staph aureus infections, deep surgical site infections, you can see the relative risk at the bottom of the slide was 0.21. That represents an almost 80% reduction in deep surgical site infections due to Staph aureus. So pretty dramatic result. This was followed by a more recent study that was actually coordinated out of University of Iowa here, where Maren Schweitzer and her colleagues coordinated a 20-center observational study, intervention study, involved over 42,000 surgeries. So this was really a pragmatic study to look at sort of proof of concept. Is the intervention that was done in this randomized controlled trial, if you do it across a large healthcare system, can you also see a similar reduction? And sure enough, when they screened for Staph aureus and then applied mupirocin and chlorhexidine decolonization, they were able to reduce complicated Staph aureus surgical site infections by over 40%. And the red box to the right is basically just to show that they were able to reduce surgical site infections due to both methicillin-resistant Staph aureus, or MRSA, as well as its more susceptible cousin, methicillin-susceptible Staph aureus. So why did this large pragmatic study show only about a 40% or slightly over 40% reduction compared to the almost 80% reduction in deep surgical site infections that was shown by Bodie and colleagues? Well, this really has to do with the difference between a randomized controlled trial and an actual intervention in a hospital, which requires really good implementation. Because if you look at all of the hospitals and the patients involved in this study, in the group, and it's about 40% that were fully adherent to the protocol, in other words, got every aspect of the decolonization protocol, the relative risk, you can see at the top, relative risk was 0.26, so an almost 75% reduction if they did everything that was in the protocol. So very similar to the 80% reduction seen in the Dutch study. There was less impact if you were not able to adhere to all the elements, like maybe they didn't get more than a day of mupirocin, or they did not get the chlorhexidine, et cetera, et cetera. So after looking at these data, you might say, well, why don't we just find everyone who carries Staph aureus and try to decolonize them? That would be one sort of public health intervention to try to reduce the rate of Staph aureus infections. Well, the drawbacks of that are, first of all, decolonization is often transient. It's very difficult to decolonize someone permanently. Up to 40% or more of patients that you give Staph aureus decolonization will recolonize within about 90 days of the decolonization step. So it's really best performed right before the high-risk period. So in a surgical patient, for example, decolonizing them immediately preoperatively so that you carry them through that high-risk period for surgical site infection, and you may not, then by the time they recolonize, their surgical wound is healed and they're at lower risk of infection. Secondly, if you were going to try to track down all Staph aureus carriers and decolonize them, that would be a huge and really complicated and impossible intervention, because there are over 80 million people in the US by now that are colonized with Staph aureus. And then finally, anytime you widely use an antimicrobial, if we've learned anything by now in infectious disease and microbiology, it's that exposure to an antimicrobial agent will inevitably eventually lead to resistance. And the compound we use currently most often to decolonize staph in the nose is mupirocin. And we know from going back more than 30 years to some of Carol Kaufman's work in 1993, they showed that when they tried to use mupirocin in a long-term care facility, after widespread use, the rate of resistance jumped to 11%. And most recently, Mary Hayden and her colleagues reported from a very large study that used mupirocin and chlorhexidine called the Reduced MRSA study. They found that there was about 8% high-level mupirocin resistance among MRSA in that study. And this is concerning because high-level mupirocin resistance, as many of you may know, is plasmid-mediated, so it can spread very rapidly. Chlorhexidine resistance is also something to be concerned about. In this study, they found a very low rate of detectable resistance in very few of the mutations that we associate with chlorhexidine resistance. However, I think we're still learning, how to assess for chlorhexidine resistance. There's really no standard approach to doing susceptibility testing. So these reasons, the transient nature of decolonization, the widespread nature of Staph aureus carriage, and the possible emergence of resistance, those are reasons why we don't try to seek out every staph carrier to decolonize them. So now let's move from the nose to the microbiome of the skin. This paper by Philip Price was one of the early investigations of the bacteriology of normal skin and the disinfectant action of trying to clean or cleanse the skin. The image is not from that paper. That's a scanning EM of the skin surface. And the reason I show this paper is because the concept in this paper is one that I still use when I think about the skin microbiome, and that is I divide the flora into the microbiota of the skin into so-called resident flora and transient flora. And by resident flora, we mean those organisms that are present all the time. They're very adherent to the skin surface, maybe live under the stratum corneum. These are organisms that are relatively low virulence, like coagulase-negative staph, including Staph epi, cariniform bacteria, or diphtheroids. And these are felt to be protective, part of the healthy microbiome of the skin, and generally non-pathogenic. Then by transient flora, we're talking about organisms that are more superficial, probably more amenable to removal with disinfectants and cleansing, and are generally acquired by direct contact with a surface or another person or other fomite that carries the organism. So they're more transmissible. And in the hospital setting in particular, we think about Staph aureus and MRSA, gram-negative bacilli, Candida, and other organisms that we sometimes see transmitted from one patient to another. We consider those mostly to be transient flora. And so one of the ways that we try to reduce the risk from transient flora or more pathogenic transient skin flora is, again, using chlorhexidine and basically bathing patients in the hospital every day with a 2% chlorhexidine gluconate solution. There have been a lot of, again, observational before-after studies that have looked at chlorhexidine bathing. I'm showing you this study because it happened to have been a randomized trial, a cluster randomized crossover trial in 9 intensive care or bone marrow transplant units in 6 hospitals. The intervention was to bathe every day the patient with 2% chlorhexidine gluconate versus bathing with bland soap. And the primary outcomes were MRSA and VRE acquisition in the study group and hospital-acquired bloodstream infections. And the reasons for these primary outcomes is because those are the outcomes that had been demonstrated to be improved in the observational studies done prior to this randomized trial. And so I'm just showing you quickly the results in this study. That bar graph is the rate of bloodstream infection per 1,000 patient days. In the control group in dark blue and the light bars are the chlorhexidine group. And you can see that bloodstream infection rates were reduced by almost 30%. Now, many of these infections were those due to coagulase-negative staph, which is a lower virulence organism, not Staph aureus or gram-negative organisms. But they also reduced MRSA, methicillin-resistant Staph aureus, and vancomycin-resistant enterococcal acquisition by almost a quarter, which was statistically significant. MRSA reduction by about 19% and VRE reduction by about 25%. So this trial basically confirmed the benefit of daily chlorhexidine bathing that had been shown in other previous observational studies. And it's a very simple intervention that most hospitals are now incorporating, at least in the ICU environment, to reduce the acquisition of, well, MDRO, a multiply drug-resistant organism, including MRSA and VRE, and in an attempt to reduce bloodstream infections, primarily due to skin organisms like coagulase-negative staph. Now, again, we have to consider the limitations of this approach, including the possibility, again, of resistance to chlorhexidine, or there are some organisms that are inherently less susceptible to chlorhexidine, and will we start seeing increased rates of carriage due to those organisms? I think we still have a lot to understand about the potential implications of widespread use of chlorhexidine bathing in the hospital. So before I move on to the gastrointestinal tract, I want to at least mention another way that we use chlorhexidine to alter the human microbiota in hospitalized patients, and that is daily oral care with chlorhexidine in those patients who are being mechanically ventilated. This is now routine in ICUs around the country and around the world as part of the so-called bundles to prevent ventilator-associated pneumonia. And it's based upon the observation that most ventilator-associated pneumonias are felt to be due to organisms that first colonize the oropharynx and then through microaspiration events are introduced into the lower respiratory tract. And if you can decrease the microbial burden and really alter that abnormal microbiota in the hospitalized patient, you may be able to reduce the risk for ventilator-associated pneumonia. There have been a lot of studies looking at this. I'm just showing you one meta-analysis published now, quite a little while ago, actually. It wasn't recent. But the COLA meta-analysis is where the forest plot on this slide comes from. And you can see that there was, when they looked at 12 different randomized controlled trials, in one of these meta-analyses in Lancet ID, they saw about a 30% reduction in risk for ventilator-associated pneumonia. So yet another use of chlorhexidine to alter the microbiota. So now I want to spend the rest of the time talking about the microbiota of the gastrointestinal tract. So the GI tract is probably the richest bacterial reservoir in the human body. Up to 100 trillion bacteria colonize the human gut, up to 1,000 distinct species. And the sort of composition of a healthy microbiota in the gut is felt to be protective. And it's felt to be protected by providing what we call colonization resistance. So the presence of this diverse microbial community protects the host from particular pathogens that, in the absence of this community, may have an opportunity to cause disease. A healthy microbial community in the gut is very likely to have many other implications for human health, which we are just now starting to understand. But I'm going to focus primarily for the rest of the time on Clostridium difficile, which is an anaerobic gram-positive organism that colonizes the gut in many people, but it can also cause disease, particularly when the microbiota of the gut is disrupted. So why should we be talking about C. diff infection? Well, I would say that in the US and in most other parts of the world, Clostridium difficile infection may be the most important healthcare-associated infection currently. There are increasing rates worldwide. The increase that occurred in the 2000s, which is illustrated on this slide from the National Center for Health Statistics, was really quite astonishing. These are deaths per million population due to C. diff infection. And you can see from this slide that it basically quadrupled over a 10-year period of time. And not only are we seeing more deaths due to C. diff infection, we're seeing more severe disease, more treatment failures, more folks who need colectomies because of C. diff, and we're even starting to see more community onset cases of Clostridium difficile infection. So it's a big problem. And it turns out that the main host defense against C. diff infection is to have an intact microbiota in your colon. There's clearly some role for humoral immune response as well, can be protective, but the intact microbiota is really the key here. So in every patient with C. diff, or virtually every patient with C. diff, has had these two events occur: exposure to an antimicrobial agent that's disrupted their microbiota, and associated with either exposure to or antecedent carriage of Clostridium difficile that is toxigenic, that is capable of producing the toxins that cause disease. So what is the disease we associate with C. diff? What are the clinical manifestations? Well, as I mentioned, a percentage of us carry Clostridium difficile as part of our microbiota. In many cases, toxigenic C. difficile, but we never develop any symptoms or disease. It's felt to be present in 3 to 7% of healthy adults, up to 15% of newly admitted hospitalized patients, and even up to 50% in some studies of long-term care facility residents. Most of the time, it causes mild to moderate diarrhea and is eminently treatable, responds to therapy in 85 to 95% of the time. The big problem being recurrent infection, as we'll talk about. But the more severe cases involve a diffuse colitis called pseudomembranous colitis, and you can see on this picture the exudates that are present on the surface of this inflamed colon. This can lead to bowel perforation, septic shock, and death. So as I mentioned, a big problem with C. diff infection is not necessarily the treatment. As I said, well over 85 to 90% of patients respond to therapy with reduction or resolution, complete resolution of the diarrhea. They feel better. The problem is the recurrence rate, which is exceedingly high. The risk of a first recurrence after you have C. diff infection is about 25 to 30%. Those who had a recurrence and then are treated have an almost 40% chance of a second recurrence, and then another 30% of a third recurrence and a fourth recurrence. This is felt to be due to the fact that C. diff, as you know, exists in a spore form, which is very resistant to antibiotic therapy, or really impervious to it, so that perhaps spores can then result in regrowth of vegetative bacteria after treatment is completed and the patient goes from feeling well again to having C. difficile. Many of these recurrences require hospitalization, and many have severe consequences. So why do people have recurrent disease? Well, it's because when you disrupt the gut microbiota with antibiotic therapy, it does not return to normal immediately. If it did, you could treat the C. diff, stop treatment, and then expect the gut microbiota to basically reconstitute very quickly and be protected. It varies from individual to individual, but we do know from some fairly well done studies now that those patients who go on to develop recurrent C. diff infection or frequent recurrences are those patients who have very slow return or no return to a healthy, diverse microbiota. Whereas those who don't have recurrences are those who've been able to reconstitute this diverse microbiota. And some studies have shown disruption of microbiota for up to 2 to 4 years after treatment with certain antibiotics in some people. So this disruption of the gut microbiota is long-lasting, which is really why we have such a problem with recurrent disease with C. difficile. So this is where fecal microbiota transplantation, or some call it fecal microbiota transfer, we'll shorten that to FMT for the purposes of this talk. That's where this comes into play. The rationale being that if the fundamental problem with C. diff infection and recurrent C. diff infection isn't necessarily just the presence of C. diff, but the absence of healthy flora to keep the C. diff from causing disease. So the goal of this is not to eradicate necessarily the C. diff, but it's to reintroduce a stable community, a diverse community of gut microbiota that disrupts this, or that replaces the disrupted native microbiota. So there's a long history, actually, of FMT in the veterinary field. Anyone who's owned a dog, for example, knows that animals often consume their own feces or other animal species. This has always been felt in the veterinary world to perhaps contribute to the development of the GI tract and be actually a healthy or adaptive behavior. This has also been used in veterinary medicine for the treatment of certain gastrointestinal conditions. In the vet world, they call it transplantation, from what I understand. And so they were sort of way ahead of at least modern human medicine in picking up on the utility of fecal microbiota transfer. So in terms of human use of FMT, there are some descriptions in the Chinese literature going back to 300 AD, describing using human fecal suspensions for those who have gastrointestinal conditions, felt in some cases to be a quote-unquote miracle cure for those patients. There's a reference in the German literature to the medical uses of human and animal feces, and references to observations in World War II of Bedouins in northern Africa ingesting camel dung to treat dysentery. So these are all sort of pre-modern medicine uses of fecal transfer to treat disease. So the first sort of what I would call modern era of medicine publication on FMT was published in 1958 in the journal Surgery by Dr. Eisman. He describes performing stool transplants, and he did this by taking stool from healthy donors and delivered it via fecal retention enemas to 4 patients who had fulminant pseudomembranous colitis. So if you remember that photo with the exudative inflamed areas of the colon, that's what we call pseudomembranous colitis. At that time, it was not known that this condition was due to Clostridium difficile. It was known that it had a very high mortality, and it was also known that it was associated with prior antibiotic treatment. So all 4 of these patients improved and were discharged home after this treatment. So interestingly, it really, this didn't stimulate at that time significant ongoing interest in FMT. So it really, if you flash forward now, over 50 years before we got the first real randomized controlled trial of this approach to treat recurrent, in this case, recurrent C. diff infection. And this graph is from the Nude Study in the New England Journal of Medicine, a very influential study that brought, I think, FMT from being sporadically offered in different centers to a more widespread practice for the treatment of recurrent C. diff infection. What these investigators did was a randomized controlled trial of 3 treatment arms. The first was the fecal transplant via a nasoduodenal tube, and this was done after the patient had received 4 days of vancomycin and polyethylene glycol bowel lavage. The other 2 groups were vancomycin 500 milligrams 4 times a day for 14 days. One of those 2 groups also got the polyethylene glycol bowel lavage. This study was terminated early because of how effective the fecal transfer was. As you can see from the graph on the far left, an 81% cure rate, which means no relapse within 10 weeks of the infusion of donor feces. And then the cure rate went up to 94% with a repeat for those who weren't cured with the first go-round. And you can compare that to only a 23 to 31% rate of cure without relapse in the vancomycin group. So this is supported by many other case series, another randomized controlled trial. And if you are interested in sort of the aggregated data. You can look at this Annals of Internal Medicine paper by Diekem and colleagues from last year. They looked at 28 case series, 2 randomized controlled trials, over 500 patients, and the overall cure rate was about 85%. So a highly effective approach to treating recurrent C. diff infection. I'm showing you this paper because I think it's an example of the often really dramatic response you see to FMT, and also the really sad stories that you hear if you're an ID clinician in practice, as I am, with patients who have recurrent C. diff. So this was a 61-year-old woman with over 8 months of diarrhea, just a horrible situation, bowel movements every 15 minutes, having essentially to wear a diaper at all times, had lost 60 pounds, and failed multiple courses of therapy to try to keep this infection at bay. Well, she underwent an FMT from her husband, and within a day or two had her first solid bowel movement. Within a month was actually constipated and had not had a recurrence of C. diff infection at 6 months. And then if you look at the graph on the right from this, the metagenomic look at her fecal microbiome compared to her husband, the donor, you can see what happened. So the second column is the diversity of the patient on day 0 in terms of the flora in the colon. And then the donor is the third column, and then the fourth column is the patient on day 14. And you can see how similar the microbiota of the patient 2 weeks after FMT is to the donor on the day of FMT. And then day 33, you see there's still a healthy amount of Bacteroides. There's a healthy amount of the Ruminococcaceae, which is a firmicute, which is also seems to be healthy, part of a healthy, diverse colonic flora. So anyway, this I think is a very good example of the kind of individual results that you can see with FMT. So what about the downside of FMT? Are there serious adverse events associated with this approach? And indeed, there have been some reports. It's difficult in some cases, even though the frequency is very rare, to determine whether an adverse event was due to the FMT or due to the inflamed colon from C. difficile infection. For example, the 4 episodes of gram-negative bacteremia reported in this review of the literature. Baxter and Colville looked at 109 papers to determine the rate of adverse events after FMT. Perforation occurred in 3. Sometimes this can be related to the mode of delivery. As we'll discuss, some FMT is done via endoscopy or colonoscopy, which does carry a risk of perforation. And then for attributable deaths, there were 2 episodes of aspiration. One was after sedation for a colonoscopic delivery of FMT, and one was aspiration delivered when FMT was delivered via upper endoscopy. So very low frequency of adverse events, but nonetheless something to consider. And then, of course, there's the fact that we know so little about the other impacts of changing one's microbiota. This is a rather somewhat humorous headline from the Daily Mail in Britain, but it was about a case that actually was subsequently reported in Open Forum Infectious Diseases last year, and that is of a 32-year-old woman who had a fecal transplant in 2011 for recurrent C. diff infection, and got this, the donor was her daughter, who's overweight, and as this Daily Mail story reports, she went from 9.5 stone to 12.5 stone with a BMI increase of 34.5 after this. Now, obviously, this is a single case, but I don't think it's crazy to realize that there may be unanticipated consequences of completely replacing someone's fecal microbiota. In my opinion, though, the really life-saving nature in some cases of FMT outweighs those somewhat theoretical concerns at this time. So there's a lot of remaining questions about FMT, one being what's the best way to administer the fecal sample. Most often it's done via nasogastric or nasoduodenal tube or endoscopy. It can be done via retention enema. That takes a lot longer and is more uncomfortable. And there's some recent data now on using frozen capsules. And I'm going to show you this study. Some refer to these as Capsules, actually. This report from JAMA in 2014 is an open-label, uncontrolled study of 20 patients with recurrent C. diff. And they put about 650 microliters of a fecal slurry inside each of the capsules and gave the capsules daily for 2 days. And 14 out of the 20 had resolution after one treatment course without recurrence. And of 6 non-responders, 5 had resolution. 1 of those 5 did recur within the 6-week time period. But the overall response rate, 90 to 95%, was very high, and there were no adverse events. No patient vomited, thankfully. And there's been such enthusiasm for FMT that there was actually published in the Clinical Gastroenterology and Hepatology Journal, do-it-yourself home instructions for doing FMT. I do not recommend this, but just so you know, this stuff is out there, and some of your patients, if you're a practicing physician, may decide to pursue it if they don't have the availability of FMT within their area. And they can even go to YouTube and see someone demonstrating how to do this. Again, not something that I would recommend. So obviously there's an ick factor associated with FMT, and even now with the increasing use of stool banks, which is what we do here, we use a stool bank to provide a stool from screened healthy donors, and with the ability to do look backs, et cetera. It's very, it really makes it much easier than doing related donor situations. But even so, there is this ick factor. So it would be really nice if we could use a product that is actually cultivable in the laboratory and serves as a substitute for stool flora. And so this is what some call microbial ecosystem therapeutics. I'm showing you data from 2 published papers from the same group, or at least Tavetti was an author on both of these papers. In total, in this work, they present 14 patients who had recurrent C. diff infection, and they were treated with a mixture of 10 different bacterial species that are listed in the right corner there. You can see that 7 of these are anaerobes, Clostridia, Bacteroides, and then a couple of strains of E. coli and Enterococcus. They were grown in pure culture, then combined with sterile normal saline and given via enema. Over 86 or 85% or 86% response rate. Patients became asymptomatic and no longer had detectable C. diff toxin. This is another paper looking at the same general approach. This was really just a report of 2 cases in the journal Microbiome from 2013. What they did was they took donor stool from a 41-year-old woman and cultured 62 different bacterial isolates from the stool, including culturing under strict anaerobic conditions. They chose 33 strains based upon their susceptibility. In other words, if they were going to infuse these organisms into the GI tract, they wanted to use only those that were not multiply resistant. So they chose more susceptible species, and you can see them all listed on the right there. And they mixed these in a ratio that was designed to try to mimic the metagenomic database of stool samples from healthy donors. They took this final product, which they called Repopulate, and delivered it via colonoscopy to 2 patients with recurrent C. diff infection, and both remained symptom-free at 6 months. So there are other published studies looking at, lab cultivable stool substitutes. I just picked two somewhat at random to show you some of the literature that's out there. Another approach I thought was interesting was using bacterial spores to treat recurrent C. diff infection. In this study by Kana, which was just published in the Journal of Infectious Diseases, they enrolled 30 patients who had 3 or more C. diff episodes in this open label study. And what was involved was taking donor stool from 7 healthy donors, suspending it in saline, filtering it, exposing it then to 100% ethanol to kill all the vegetative bacteria and leave only the spores. And the spores were from firmicutes, as you might imagine, 50 different species. They were placed into capsules. The capsules were frozen, and then they were delivered in 2 different dosing strategies to these patients and had a very good rate of overall clinical resolution at 8 weeks. So another interesting approach using bacterial spores to try to reconstitute a healthy colonic microbiota. And then finally, I want to mention a paper that's gotten a lot of attention from Dale Girding and his colleagues in JAMA last year. And this was a phase 2 double-blind randomized controlled trial using non-toxigenic C. difficile spores to treat C. diff infection, either the first episode or the first recurrence. And so you're really trying to replace potentially toxigenic C. diff, and into that niche, you want to establish a non-toxigenic C. diff community. And so they had 4 groups here, a placebo group of 40 patients. Placebo group had a C. diff recurrence rate of 30%, which is typical. And then they had 3 dosing strategies for the non-toxigenic spores. Overall, the recurrence rate was half, or 14%. And interestingly, if you take all of those patients who had received the non-toxigenic C. diff spores, those that established colonization with non-toxigenic C. diff via subsequent culture had an only 2% CDI recurrence rate. So I think this is also a promising approach to treating recurrent C. diff infection. Finally, I want to mention another very interesting area for manipulation of the colonic microbiota, and that is to reduce or eliminate the carriage of antimicrobial resistant bacteria. This study, which was published this year in Clinical Infectious Diseases by Milan and colleagues, looked at what happens to the number of antimicrobial resistance genes before and after a FMT in patients who have C. diff and healthcare exposure. And you can see from, in this study, what they did was they collected stool from the donor and the recipients both before and after, and at time intervals after, 2 to 4 weeks, 4 to 8, and 8 to 22 weeks. And then they took the metagenomic data and aligned it to the comprehensive antibiotic resistance gene database. And what they saw, you can see on this table, on the far left are the donors, very few antibiotic resistance genes per person. The BFT column is before fecal transplantation, and you can see it varied by subject, but there were some who had a lot of antibiotic resistance genes, probably because they had been exposed to antimicrobials, which is why they got C. diff. But after FMT, you can see a really dramatic reduction in the number of detectable antibiotic resistance genes present in the fecal microbiota. So I think this is very promising and maybe a harbinger of another use of microbiota manipulation. So that's really all I have before I take questions. In summary, the human microbiome obviously plays key roles in health and disease. We're just really scratching the surface of how we manipulate it to prevent or treat infection. But I think it's going to increasingly focus on microbiota alteration, trying to restore a healthy ecosystem. And currently, the approaches that are in routine use are suppression of the Staph aureus nereis carrier state, suppression of transient skin and mucous membrane microbiota, usually with chlorhexidine, and replacement of disrupted gut microbiota. I'm hopeful, though, that future approaches will be able to target the microbiota much more precisely, and I think some of the early work we're seeing on replacing the gut microbiota with something slightly less crude than feces, you know, a lab cultivable solution, I think is where we're heading. So with that, I will take questions.
Thank you very much, Doctor, for that wonderful presentation. We do have a couple of questions here, and for our audience, if you are still wanting to submit some questions, you would be able to do so by pressing the green Q&A button on the lower left-hand corner of your screen. The first question that comes in is why microbiota that becomes pathogena, or for example, Staphylococcus aureus.
I'm not sure I understand. I'm not sure I understand the question. Can you repeat it?
All right. No, thank you very much. I appreciate that for the clarification. So essentially the question is, why does microbiota become something to the example of Staphylococcus aureus?
Okay, I think I know what the question is. So I think this question was how is it that a member of the normal flora, like Staph aureus living in your nose, how does that become pathogenic? What are the circumstances that allow that to occur? And in general, what we think is that Staph aureus that is living harmlessly in your anterior nares doesn't cause a problem until you have some kind of a disruption in normal skin or mucous membrane barriers. And then because Staph aureus in the nose often transiently has access to the skin, if, for example, you have a surgical wound or you have a dialysis catheter or for some other reason you're immunocompromised, that organism can then gain access and invade and then cause infection. So that's the reason for, in the case of Staph aureus, that we think it can move from being a commensal to a pathogen. And then what I'll do is I'll just read a couple of other questions. So one is, I heard that some people with inflammatory bowel disease, like ulcerative colitis, have used fecal microbiota transplant with some success. Is there any good data to support that practice? So I think that's a really good question. There are some folks with inflammatory bowel disease who really do swear by fecal transfer, FMT, to treat their disease. But I'm still waiting for better data, and I think most of us are, to suggest whether that is in fact effective in inflammatory bowel disease, and I believe that some of those studies are going on now. So then there's another question here. Is there reason to believe that certain antibiotics are better than others in terms of how they impact the microbiome of the gut? And does that go into your decision-making about antibiotic choice? So that's another really good question, and an area where we really need more data, because as those of you who practice in clinical settings know, antimicrobial stewardship is becoming an absolute key intervention to reduce antimicrobial resistance and improve outcomes. And one of the things that we don't yet know is what is the hierarchy for antibiotics in terms of how much they disrupt the microbiota. And if that could be factored into our stewardship approach and to our treatment guidelines, perhaps then we would see fewer cases of C. diff, for example, if you had a treatment guideline that said, here's 5 ways you can treat community-acquired pneumonia, but we are going to move this one up because it appears to not have as much of an impact on the microbiota of the colon. That would be a great situation. So I think right now we need a lot more studies to look at that. And let me just take one more look and see. I think we've pretty much dealt with the questions that have been asked. So if that's everything, then I will turn it back for a wrap-up.
Well, thank you very much, Doctor, for going through those questions with us today. I guess the final question would be for you, is there any last comments that you would like to leave us today?
Well, I think the main comment I would make is that we're really just scratching the surface in terms of the role of the human microbiome in human health and disease, and the approaches we're taking right now to manipulate the microbiota are really quite crude. And I'm really hopeful that through further research, which requires further funding, to improve our understanding of the microbiota, will improve our ability to be more, as I said, more precise in our approach to preserving a sort of healthy, diverse microbiome.
All right. Today's webcast will be available for online viewing on our website through December 7th, and we'll let you know when it becomes posted. And we hope that you pass this information along to your colleagues who are unable to view today's presentation. We'd like once again to thank the fine doctor and Lab Roots for this wonderful presentation time. And see you next time. Goodbye.